All right, now since we have a 7-part video already posted on YouTube that we've asked you to watch, I'm going to go through this very, very quickly. So, cellular respiration, what is it? It is basically the process by which the energy that is contained in organic molecules can be released. And that energy is then used to attach phosphates onto ADP and form this gasoline called ATP. So fundamentally, the whole purpose of cellular respiration is to produce this gasoline called ATP. So the analogy that I gave, and this is in the video, but I'll just remind you of the same analogy that I presented. I gave the example that what's really cellular respiration, the purpose of what's really going on, is very much like a change machine at a laundromat, at a coin-operated laundromat. If you've ever been to a coin-operated laundromat, they have washing machines and dryers, and it seems that they almost always run on quarters. Does anybody know what I'm talking about? Yeah. All right. I don't know who decided they were going to take quarters, but that's how they are. So usually if you want to wash your clothes, you need 2 quarters or something like that. You need to dry them, use a quarter. So let's imagine that you've got a $10 bill. So what you have to do is you have to go to a change machine that they usually have in the laundromat. You stick the $10 bill in, and since $1 is 4 quarters, $10 is 40 quarters. Now you've got quarters, so you can run the machines. Is everybody okay on that? So how does this relate to biology? For some reason, everything in biology runs on these gasoline molecules called ATP. But what if you have a sugar molecule? A sugar molecule is like a $10 bill. And what you have to do is convert the energy that's in that $10 bill or sugar molecule into a bunch of smaller value quarters called ATPs. So just like a change machine converts a $10 bill into 40 quarters, cellular respiration converts the energy in sugar into about 36 or 38 ATP molecules. Not quite 40, but similar. Now, for some reason, just like all the washers and dryers run on quarters, all the chemical reactions and all living things run on ATPs. So, the question is, how do we release that energy that's in the sugar and turn it into ATPs? So in this overall process, this overall reaction is an oxidation-reduction reaction. We have a glucose molecule. We're going to combine it with oxygen. There are a whole bunch of enzymes and coenzymes. This really is a multi-step process. It's not a single step. And eventually it forms 6 carbon dioxides and 6 waters. And that energy that was contained in the sugar is released. And that release of energy is used to attach phosphate onto ADP and form ATP. The name of the process of attaching a phosphate onto ADP to form ATP is called phosphorylation. We originally learned that term back in section D. Now, about 40% of the energy that's released from breaking apart the sugar into carbon dioxide and water is used to phosphorylate ADP into ATP, and it can result in the production of a maximum of about 38 ATPs. Most books will say 36. It frankly varies depending upon the cell, whether it produces 34 or 36 or 38 ATP, but the maximum is about 38. It's close to the idea that a $10 bill gives you 40 quarters. A glucose molecule gives you close to 40. It's about 38. But in fact, last time we lectured, or the time before that, we learned, a couple of lectures ago, we learned that about the first and second laws of thermodynamics. And we learned that whenever you try to convert one form of energy into another form of energy, some of that energy goes off as heat. We explained in the case of a light bulb, you're converting electrical energy into light energy, but a light bulb gets hot. There's no way to have 100% perfect conversion of electrical into light. You're always going to get some heat. So similarly, even though we're trying to convert the energy that's in sugar into ATP, in fact, over half of the energy, 60%, goes off as heat. Now, looking at the details here, basically what's going to happen is that this glucose molecule, this sugar molecule, is going to be split apart in half, and then each of those halves is going to be broken apart, and we're going to keep breaking it apart into smaller and smaller pieces until all that's left of it is carbon dioxide molecules. So here it shows that glucose is going to be oxidized into carbon dioxide. Now, we know that an oxidation reaction is a loss of hydrogens. So as this sugar is being split apart and the hydrogen atoms are being broken off it, incidentally, that splitting of these covalent bonds that hold these atoms together is what's releasing the energy, because that energy is contained in these bonds. Remember, it takes these covalent bonds are very strong bonds, and breaking them releases a lot of energy. So anyhow, what happens to the hydrogens that are broken off this sugar molecule? The answer is there's a coenzyme called NAD that picks up these hydrogens two at a time and eventually transfers them to oxygen. NAD actually contains the B vitamin niacin. So the role of niacin, or NAD, is to actually transfer hydrogens from the sugar molecule ultimately into oxygen. When the hydrogens are transferred to oxygen, that turns it into water. All right, so we say that oxygen is chemically reduced into water. Because what does reduced mean? Reduction is a gain of hydrogens. So oxygen is gaining those hydrogens. So glucose is oxidized and oxygen is reduced in this oxidation-reduction reaction. And so attaching the hydrogens on oxygen forms water. Now, the question is why? Why couldn't we simply have broken apart this sugar and broken off those hydrogens? Why can't we just break apart those hydrogens off the sugar, release the energy, and make ATP? Why did we have to transfer the hydrogens onto oxygen? Think about it. If you break the hydrogen atoms off a sugar molecule and release these hydrogens either inside the cell or even surrounding the cell, what does that do to the acidity inside or surrounding the cell? What is the definition of acidity? Hydrogen atoms. So if you break these hydrogen atoms off the sugar molecule to release the energy that's contained in the sugar, that would increase the acidity. So the way living things solve this problem is they transfer those hydrogens that were broken off the sugar onto oxygen. That's why we breathe oxygen. Because transferring the hydrogens onto oxygen forms something totally harmless to a living thing called water. That's why we're breathing oxygen. So in fact, we wrote in bold print, in a large font size, the reason why we breathe oxygen is that it acts as a hydrogen acceptor. It accepts the hydrogens that were broken off the sugar molecule to release the energy that was in the sugar in order to make ATP. All right, now again, everything I'm covering, you can watch on videos over and over again. So, cellular respiration, we wrote, is the process by which organic compounds, usually sugars, like glucose, are broken apart, releasing energy that is used to produce ATP. This process of cellular respiration occurs in all living things. Not only do animals, including humans, convert the energy that's in sugars and fats into ATP, but so do plants. So this is universal. This is universal. We had learned the difference, one of the differences between plants and animals is how they got their organic molecules, how they got their food, how they got their sugars. Plants can make their own foods, their own sugars. We have to eat them. But all living things, once they have those sugars, once they have those fats, they have to take the energy that's in those sugars and fats and convert them into ATPs. Just like a change machine converts a $10 bill or a $20 bill into quarters. So that's what we wrote here. Now, we divide cellular respiration into two phases, anaerobic and aerobic. The anaerobic reactions literally means without air, without oxygen. And these reactions occur in the cytoplasm of a living cell. The aerobic reactions, and the word aerobic means with air, with oxygen, these reactions occur in the mitochondria of the cell. Now, before we examine this in more detail, I want to first address the whole issue of sugar. Because obviously, if we and all other living things are breaking apart sugars and converting them into ATP, how does our body ensure that we've got enough sugar to convert into ATP? To address this subject, let's look on the next page, H2. And on H2, here's our question on H2. How are the glucose levels, the sugar levels, regulated in our bloodstream? Because obviously the cells need enough sugar so they can convert those sugars into ATP. The process of converting sugars into ATP is called cellular respiration. But before we are going to address that, we first want to address this earlier question: how does our body ensure it's got enough sugar so it can convert them into ATP? There are two very important hormones that regulate our blood sugar level. One of them is called insulin, and the other is called glucagon. Insulin and glucagon. Now, I wrote here on the left that both insulin and glucagon are protein hormones. We know that hormones fall into two categories: protein hormones or steroid hormones. And back in section D, we gave you examples of steroid hormones, including testosterone or estrogen, made from cholesterol. And also in section D, page D5, we talked about examples of protein hormones, including insulin and oxytocin and growth hormone. Both insulin and glucagon are protein hormones. And both of these hormones are made in your pancreas. Again, we had actually talked about insulin back on page D5. What does insulin do? And what does glucagon do to regulate our blood sugar level? We wrote that insulin causes a process called glycogenesis. Glycogenesis. I know you'd say, I can't read that because you made a mess of it. All right, well, we'll see that word written better in a moment. But it's a very good word. It has the word glycogen here. Glycogen is the polysaccharide that is stored in our liver cells. Genesis means to make, to form. So literally, it means what glycogenesis is that insulin causes the sugar in our bloodstream, the glucose sugar in our bloodstream, to be transported into our liver cells and other cells and stored as a polysaccharide called glycogen. So I represented glucose by drawing one red ball because it is a monosaccharide. And glycogen is just a whole bunch of glucoses joined together. It is a polysaccharide represented by a whole series of red balls. Now, if insulin is causing sugar in our bloodstream to go into cells, including liver cells, and causing those glucose molecules to be joined together into this polysaccharide, it's a way of storing sugars for when we don't really need it. When you're eating a meal and you're relaxing and you're not using energy at a high level and you're absorbing all this sugar from your meal, then insulin is released from your pancreas and causes the sugar in your bloodstream to be stored in your cells, including your liver cells, as glycogen. So what does insulin do to the amount of glucose in your bloodstream? Is it increasing it or decreasing the amount of glucose in your bloodstream? It's decreasing it. It's causing the glucose in your bloodstream to enter the cells and be stored as glycogen. It's decreasing the amount of sugar in your blood. This is exactly what we told you back in page D5. Now, glucagon does the exact opposite. You could think of these two hormones as the yin and yang of each other. They're exactly the reverse. Glucagon causes glycogenolysis. Very good word as well. We see the word glycogen, and we see the ending lysis. Lysis always means to break apart. So glucagon is a hormone secreted also by your pancreas that causes the glycogen in your liver cells to be broken apart into glucose and released into the bloodstream. So here's how I represented it. Glucagon hormone causes the glycogen, represented by this chain of red balls, because it's a polysaccharide, representing glycogen, to be broken back apart into glucose and to release this monosaccharide glucose into the bloodstream. Let's show you one more picture to help you visualize this. Let's look on page H8. H8. And on page H8, so on the very top of H8, this is showing a liver cell. It says liver cell on the top of H8. And here's what we tried to represent. It shows that when we have glucose in the bloodstream, glucose is a monosaccharide, and we don't need all this sugar in our blood at that moment. Insulin causes the sugar in our bloodstream to go into cells, including liver cells, and it causes glycogenesis. That's what's written there. It causes that sugar to go into cells and for that glucose to be joined together into this polysaccharide called glycogen. Insulin also makes sugar go into other cells in our body, and when it goes into other cells, like let's say brain cells, in that case, the insulin causes the glucose to go into the cells where it's broken apart into carbon dioxide and water and turned into ATP. But in any case, insulin is causing sugar to go into cells, lowering the amount of sugar in our bloodstream. The hormone glucagon and also the hormone epinephrine, which also goes by the name adrenaline. Both glucagon and epinephrine or adrenaline cause glycogen that is stored in our liver cells to be broken back apart into glucose and released into the bloodstream. That process is called glycogenolysis. So these hormones raise the amount of sugar in our blood because they're causing the stored carbohydrate, the glycogen, to be converted back into glucose and released into the bloodstream. So whereas insulin is used to transfer the sugar from the bloodstream into the cells, glucagon and epinephrine is used to return that sugar back to the bloodstream and make it available in the bloodstream. Let's also look at this diagram in the lower half of page H8. This is basically just summarizing what happens when we're absorbing nutrients from our food in our gastrointestinal tract, our GI tract. So when you've eaten a meal, and if you haven't yet eaten lunch yet, then you're probably waiting for this class to end so you can. So when you've got food in your digestive tract, you're going to digest carbohydrates into simple sugars, monosaccharides. You're going to break apart fats into fatty acids. You're going to break apart or digest proteins into amino acids. And that's what you actually absorb into your bloodstream. This represents the bloodstream. So here, what does it show? It shows carbs have been absorbed as sugars. Fats have been absorbed as fatty acids into our bloodstream. And proteins have been absorbed as amino acids. What happens to these nutrients as they're absorbed into the bloodstream? So you'll notice that some of the sugar circulates in our bloodstream. And in most cells of the body, this sugar is made available to the cells so they can oxidize that glucose. You'd say, what does that mean? Oxidation is a loss of hydrogens. That means breaking apart the glucose by removing the hydrogen atoms from it, breaking it apart into carbon dioxide and water for the purpose of producing these gasoline molecules called ATP. This is known as cellular respiration. So glucose is needed for cellular respiration, which is the process of converting the energy and sugars into this gasoline called ATP. You can see the sugar also provides energy for muscles and so on. Now you'll notice that in the case, though, of the liver, glucose enters the liver cells, and it tends to be stored as this polysaccharide called glycogen. We first talked about glycogen way back on page D3. The glucose can also be stored as glycogen in our muscle cells. We had learned way back on page D3 that we store, animals, including us, store sugars as a polysaccharide called glycogen, primarily in our liver and in our muscles. It's right in your notes on D3. We call it a liver glycogen and muscle glycogen. Now, just to complete this chart, you'll notice the amino acids that are absorbed from the food that we eat, they can be used by our muscle cells. And in fact, they can be joined, these amino acids can be joined together to form a polypeptide or a protein, including myosin. Myosin, we learned on page D5, is a muscle protein. Myo means muscle. These amino acids can also be used by our liver cells to manufacture plasma proteins. We learned way back on page D5 that our liver, it manufactures most of the proteins that circulate in our bloodstream, including albumin and other proteins that are made in our liver. So these amino acids are used to manufacture proteins in cells. What about the fatty acids? The fatty acids, of course, are largely stored in our fat cells. Fat cells are sometimes called adipose tissue, and we've learned way back earlier in section D that 3 fatty acids can be joined to glycerol and stored in our fat cells as triglycerides. So this is simply showing that when we eat a meal, what happens to the sugars we absorb, what happens to the fatty acids we absorb, what happens to the amino acids that we absorb. These are the nutrients that we have learned about. All right, so we have been describing how our blood sugar level is regulated. Let's just show you one more diagram. It's repetitive. It doesn't add really anything. But let's look on H11, a scary looking diagram. On H11, the only thing I want to draw your attention to for the moment, yes, we're coming to it. On H11 is it shows up here, it says proteins on the top left on page H11. We are on page H11. Proteins, carbohydrates, and lipids. Now, it could be that some of you have been here the whole semester and still don't know the difference between these 3 things. I would encourage you to go back. Remember, the final is comprehensive. There's no way you can understand biology if you don't know the difference between a carbohydrate, a fat, and a protein. And we devoted a lot of time in section D explaining the differences. But anyhow, focusing our attention right now where it says carbohydrates, and simply in summarizing, we have said that sugars like glucose can be stored as complex carbohydrates like glycogen. That process of joining glucose molecules together to form a polysaccharide called glycogen is called glycogenolysis. And the hormone that causes this to happen is insulin. Conversely, the glycogen that's stored in our cells, especially our liver cells, can be broken back apart into glucose. That process of breaking apart glycogen back into glucose is called glycogenolysis. And the hormone that promotes this process is that other hormone called glucagon. So these 2 hormones, one causing sugar to be stored as glycogen, the other causing glycogen to be converted back to sugar, are regulating the amount of sugar in our bloodstream. This is all covered in your book. Let's return back to page H1. Now that we have described the manner in which the sugars in our body, in our bloodstream, can be regulated by 2 hormones. Now, we had said on page H1, that cellular respiration, which is the name of this process for breaking apart sugars and fats in order to make ATP, which is the gasoline that powers cells, can be divided into 2 phases. The so-called anaerobic respiration reactions and the so-called aerobic. And they occur, the anaerobic occurs in the cytoplasm, the aerobic reactions occur in the mitochondria. So let's focus first on the anaerobic, because that's what occurs first. These occur in the cytoplasm of the cell. As the name says, anaerobic, these do not require oxygen. Anaerobic means without air, without oxygen. Now, we wrote at the bottom of the page that evolutionary biologists believe that this series of biochemical reactions evolved in living things when there wasn't yet any oxygen in the atmosphere. Because we have pointed out previously that we believe that the source of oxygen comes from plants that give off oxygen as they carry on photosynthesis. So originally, there weren't plants, and so without plants, you didn't have the release of oxygen into the atmosphere. Not until plants evolved did the chemical composition of our atmosphere change. In fact, the Earth is right now the only planet in our solar system that has oxygen in its atmosphere. All the other planets have gases like carbon dioxide, ammonia, and methane, but none of them have oxygen because none of them have plants to produce that oxygen. Anyhow, let's take a look on page H3. On H3, so on H3, I'm sorry, H2, apologize, H2, around the middle of the page. So the first thing that happens in the anaerobic respiration reactions is something called glycolysis. Now some of you are thinking, yeah, you just talked about glycolysis. No, I didn't. Don't confuse the word glycolysis with glycogenolysis. Glycogenolysis is the breaking apart of glycogen into glucose. That's what's shown here. Glycolysis literally means the breaking apart of glucose. So that occurs in cellular respiration. Now we've summarized that as we break apart glucose, we're going to release energy that's going to be used to make ATP. And I indicated, and we're keeping this as simple as we can, we talked about an activation step, we talked about splitting glucose in half into 2 pyruvate sugars, and so on. To help us understand this better, let's look on page H10. Page H10. Okay, now this is page H10. We're going to rotate this in the horizontal position. And this is our attempt to keep something simple, believe it or not. If you don't believe me, you're going to look at the book, because you're going to see the book goes into this in more detail. All right, so on H10, what we're seeing in this box diagram is a summary of cellular respiration. You'll notice there's a dashed line going across right around the middle of this box right here. This divides anaerobic respiration, which is shown up here, from aerobic respiration, which is down here. Anaerobic means without oxygen. Aerobic means it requires oxygen. I wrote that right here on the left. Furthermore, anaerobic respiration reactions occur in the cytoplasm. Aerobic respiration reactions occur in the mitochondria. Anaerobic are in the cytoplasm. They do not require oxygen. Aerobic respiration reactions occur in the mitochondria. They do require oxygen. So this divides these 2 series of reactions. Let's begin with the glucose molecule. That's where it usually begins. So it begins with glycolysis in the cytoplasm. We're going to represent a glucose molecule by drawing 6 carbon atoms in a row. 6 carbon atoms in a row, because after all, glucose is C6H12O6. Now we're not going to track the hydrogens and the oxygens, but we are going to at least keep track of the carbon atoms. So we start with glucose. Basically what's going to happen in glycolysis, what does the word glycolysis literally mean? It means to split apart glucose. To literally split glucose in half. So let's see that. If glucose is 6 carbon atoms big, let's simply split it in half, and that forms, and I wrote it right here, now we've gone from a 6 carbon glucose, 6 carbon sugar, to 2 sugars, each that are 3 carbons big. Does everybody see that? We have split it in half. What do we call this process of breaking a glucose, a 6 carbon sugar in half, into 2 3 carbon sugars? Glycolysis. Now the name of these 3 carbon sugars is they are called pyruvate sugars. They're called pyruvate sugars. That word I wrote here, pyruvate sugars, it's also written right here. They're also known as pyruvic acid. You'll also see them referred to as pyruvic acid. Now, even though I just simply showed taking a 6 carbon sugar and splitting it in half, forming 2 3 carbon sugars, in fact, it takes many, many steps to break this sugar apart. And we might ask, why does the cell use many, many steps just to split a sugar in half, why didn't it just break it apart in one step? In fact, why didn't we just break it, since we know eventually we're going to break the 6-carbon sugar completely apart into 6 1-carbon carbon dioxide molecules, why don't we just break apart that 6-carbon sugar, boom, 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 into 6 separate carbon atoms in one step? And the answer is, if we did it in one step, there would be an explosive release of energy. That's what happens in your car engine, where an 8-carbon octane molecule is literally blown apart in one single step, resulting in an explosive release of energy. Well, that's okay, because that release of energy is contained within these cylinders of your car engine, and your car engine is made out of steel, right? It's made out of iron, and so on. But our cells are very delicate, and you can't have an explosive release of energy where you convert glucose, a 6-carbon sugar, into 6 separate carbon dioxides in one single explosive release of energy. So the reason why there's all these little intermediate steps is so there's a controlled release of energy. A little bit is released at a time. And that's why it's many steps. All right, now, even though your book talks about all these steps, and I'm not, I am going to mention just a couple of intermediate steps. Interestingly, the very first step that leads to the splitting of glucose in half into 2 3-carbon sugars called pyruvate sugars is that 2 ATPs are needed to initiate this process. This is called the activation step. Now this is very strange, that we actually need to use the energy of 2 ATP molecules to start this process, to begin breaking apart the sugar and release energy to make ATP. Remember, what's the entire purpose of cellular respiration? To make ATP, to make this gasoline. And yet, the very first step requires we use up 2 ATPs before we produce any. So that's kind of weird, but it's not weird. In fact, we see this everywhere in nature. Let me give you a couple of examples. And I mentioned this on the video as well. What provides the fuel, the energy for your car? Gasoline. It runs on gasoline. But if that's true, and it is, why the hell do you need a battery? What's the purpose of a battery if the gasoline provides the fuel for your car? That provides the initial spark to begin using gasoline for energy. So if your gas tank is full, you got 20 gallons of gasoline, you got lots of energy in your tank, but if your battery is dead, you're not going anywhere. That battery is only needed to provide the initial spark. And once your engine starts and you're now burning the gasoline, you don't need that battery anymore. That's why if your battery is dead, all you have to do is jump start that car and your car engine's running. You just needed that battery to start the process. Another example of this is, let's say you gathered some wood to make a fire. The wood is the fuel for the fire. But what do you need to get the fire started? A match. You need a spark. You need that initial spark to begin burning the wood as the fuel. I even point out in the video, if you watched it, that this seems to be even true in the business world. Because there's a very common expression you probably have heard, it takes money to make money. The purpose of a business is to make money. But it's hard to start a business if you don't have any money. You can't even have money to make business cards. So it takes a little bit of money to start your business where the intention is to make more money. So it seems to be just a fundamental concept of the universe that sometimes it takes a little bit of energy even to eventually produce more energy. That's called the activation step. All right, once we've initiated this, this glucose is going to be split apart. I have already told you that the breaking of carbon bonds and carbon-hydrogen bonds releases energy. And that energy that's released in breaking apart that glucose in half is used to join phosphate onto ADP and form ATP molecules. What do we call this process of joining phosphate onto ADP to form ATP? Phosphorylation. We first learned that term back in section D when we first introduced you to an ATP molecule. Now you'll notice that as this energy is released and ATP are made, it shows 2 ATPs go here and 2 other ATPs go here. In other words, it looks like 4 ATPs were produced. 2 here, 2 here. Why did they draw them 2 here and 2 here? The reason why they drew it this way in this diagram is to show you that the first 2 ATPs that are produced pays back the 2 ATPs that we used up to get this process started. That just makes us break even. The next 2 represent a net energy gain. So even though 4 ATPs are produced, because it required 2 ATPs to get the process started, we're only ahead by 2. This is like the difference between when you do your taxes. They talk about your gross income and your net income. You might have grossed $22,000 last year, but after you subtract your expenses, you're only ahead by $1.37. That's your net income versus your gross income. So we grossed 4 ATPs, we're only have a net gain of 2 after we subtract out our expenses. All right, let's summarize. One more thing I should point out. I had said that the glucose was split in half into 2 pyruvate sugars, and also some hydrogen atoms were removed. You'll notice right here it shows 2 NADs, which actually contain the B vitamin niacin, have each picked up a pair of what I call hot potato hydrogen atoms. Now, the NADs are supposed to transfer those hydrogens where? Onto oxygen. Because we said you can't just release these hydrogens into the cell or even into the fluid surrounding the cell, because that would increase the acidity, because the definition of acidity is hydrogens. So these hydrogens that are being broken off the sugar are ultimately going to be attached to oxygen to form something harmless to a living thing called water. All right, let's summarize what I've just been talking about by going back to page H2. So back on page H2, we've been talking about glycolysis. What does glycolysis mean? To break apart glucose. That's literally what it means. The first step was the activation step. That's where 2 ATPs provided the initial energy to initiate or begin this process. In glycolysis, the glucose and other 6 carbon sugars are split in half into 2 pyruvate sugars. Each pyruvate sugar is 3 carbon atoms big. Furthermore, in breaking apart that sugar in half, 2 NADs each picked up a pair of what I call hot potato hydrogen atoms. Energy is released because splitting apart, breaking hydrogen atoms off the sugar, splitting the sugar apart, releases energy. And that enough energy was released to produce a net gain of 2 ATPs. We're ahead by 2. On page H3, if I can find my H3. On H3, we wrote that 2 NADs, and what's an NAD? The B vitamin niacin. We talked about it back in section F. 2 NAD coenzymes each pick up a pair of high energy or hot potato hydrogen atoms, which will be transferred to the oxygen in the mitochondria of the cell to form water. That's glycolysis. Now, what happens next depends upon whether there is oxygen available to the cell or not. To help us understand this better, let's go back again to H10. On H10, so we have formed these 2 pyruvate sugars. There they are. Now, one of 2 things is going to happen. If oxygen is available to the cell, right here it says 2 pyruvate sugars. If there's oxygen available to the cell, these pyruvate sugars are going to enter the mitochondria. They're going to enter the mitochondria, and they will undergo aerobic respiration reactions. Basically, what that means is that these two pyruvate sugars are going to be completely broken apart into carbon dioxide and water. That's what that means. But what if there isn't oxygen available? You'd say, what do you mean? Sometimes cells don't have enough oxygen available, and they can't do aerobic respiration reactions if there isn't oxygen available. So if we wrote here, if you have the pyruvate sugars, but if there's no oxygen, that's what I wrote right there, if there's no oxygen, the pyruvate sugars are reversibly converted into something called lactic acid. This process of converting the pyruvate sugars into lactic acid is called fermentation. That occurs when there's no oxygen available. Yep.
Are we talking about an anaerobic situation where there is no oxygen? Why are we saying now if there's no oxygen?
Well, sometimes there is or there isn't. In other words, we're saying if there's no oxygen, this happens. If there is oxygen, this happens. In other words, where it says pyruvate sugar, there's two arrows. One arrow is pointing this way, one arrow is pointing down. Whether it goes this way or this way depends upon whether there is oxygen or there isn't oxygen. Is that okay? Now, you'd say, well, why wouldn't there be oxygen? Well, we're going to be explaining very shortly that when we start to exercise and we start to inhaling a lot of oxygen, even though we're inhaling faster, we may not be able to provide sufficient amounts of oxygen to our muscle cells, let's say in our legs. So if our muscle cells aren't getting enough oxygen, then they can't do this aerobic respiration reaction, so instead they do fermentation and turn the pyruvate sugar into lactic acid. So we'll see why that happens. But let me basically set up the whole problem here. What's the whole problem? Here's where we left glycolysis off. In glycolysis, we split the glucose in half into two pyruvate sugars, and some hydrogens were picked up by this coenzyme called NAD. So we've got two NADs, each NAD is holding on to a pair of hydrogens. Let me pretend that I'm an NAD molecule, which is really the B vitamin niacin, and I am holding two hot potato hydrogen atoms. Now, remember, the whole reason for why you have to have oxygen is you got to do something with those hydrogens. You can't just release hydrogens into the cell or even just outside the cell, because that will increase the acidity, which would kill the cell. These are the hydrogens that were split off the sugar to release energy needed to make ATP. All right, so I'm an NAD, I'm holding two hydrogens. What I'm supposed to do is to transfer these hydrogens onto oxygen. That forms water. And that basically, water is harmless to a living thing. So that's how we inhale oxygen. We transfer those hydrogens from the sugar onto the oxygen. That forms something harmless called water. That's the ultimate goal. But what if there isn't oxygen available to the cell? Now, I'm the NAD. I'm holding the hydrogens. What am I going to do with these hydrogens? You might say, just hold on to them. The NAD can only hold on to them very, very shortly, for a brief moment in time. When there isn't oxygen available to the cell at that moment, what does the NAD do? It transfers those hydrogens right back onto the sugar where they came from. Attaching two hydrogens onto pyruvate sugar turns it into lactic acid. This is called fermentation. And the whole purpose of it was simply what to do with those hydrogens. If the hydrogens can't be transferred to oxygen, they're transferred right back onto the sugar where they came from. Let's show you that. Take a look on the bottom of page here. Yeah, the bottom of page H9. So, I'm sorry, the top of page H9. On the very top of page H9. So on the top of page H9, here it shows on your left, it says pyruvic acid, which is another way of referring to pyruvate sugar. Notice it's 3 carbons big. I'm not asking you to memorize its structure, but I want you to just look at it. It's got 1, 2, 3 carbons. Everybody see that? 1, 2, 3 carbons. Now here's the NAD. That's me. And it's holding 2 hydrogens. Since it cannot transfer those hydrogens onto oxygen, because this cell doesn't have enough oxygen available to it, we said the NAD was just going to transfer those hydrogens right back onto the sugar where it came from. So here's those hydrogens. Those 2 hydrogens are being transferred right onto this middle carbon of the pyruvate sugar. And that forms lactic acid. If you compare the structure of lactic acid with pyruvate sugar, can you see the 2 hydrogens right here? See them? I've circled them. All right? So, and you'll notice the NADH2 became NAD. So that's what lactic acid is. It is simply the pyruvate sugar that has, where it's regained those 2 hydrogens that had been removed from it. This is reversible because when the cell does have oxygen available to it again, the NAD can go and pick up those hydrogens, to reforming NADH2, and transfer those hydrogens onto oxygen to form water. So this was simply a temporary measure until the cell has oxygen again. That's the purpose of fermentation. So with that in mind, let's go back to page H3. H3. So on page H3, what is fermentation? We wrote that it occurs only when there is not sufficient oxygen available to permit the aerobic reactions in the mitochondria. There's actually 2 types of fermentation. 2 types of fermentation. There's actually 2 types of fermentation. In one type, the pyruvate sugar is turned into lactic acid, exactly as I was showing you. In the second type, the pyruvate sugar is actually turned into ethyl alcohol and carbon dioxide. Now, in the lactic acid fermentation, we wrote, this is where, because the NADs can't transfer the hydrogens onto oxygen, they temporarily transfer these hot potato hydrogens right back onto the pyruvate sugar, forming lactic acid. Exactly as we showed you in the diagram on the top of H9. And we wrote, when additional oxygen becomes available again, each NAD coenzyme will re-pick up that pair of hot potato hydrogens, reforming pyruvate sugar. Now, in which living things does lactic acid fermentation occur in? We wrote that this occurs in bacteria, and at the bottom we also wrote that it occurs in muscle cells of animals, including humans. Let's first talk about in bacteria. So in bacteria, if bacteria are breaking down sugars, releasing energy in order to make ATP, remember, all living things break down sugars and other foods to make ATP. So if bacteria are doing that, if they don't have enough oxygen, then they will do exactly as I've just been describing. They will take those hydrogens that cannot be transferred onto oxygen, reattach them right back onto pyruvate sugar, turning it into lactic acid. An example of a bacteria that does exactly that, when it doesn't have oxygen available, is a bacteria called Lactobacillus acidophilus. So what it does, this is a bacteria that lives in milk. It's actually considered a good bacteria. I mean, our first thought is, ugh, it's in the milk? Yes, that's actually considered a good bacteria for reasons I'll explain in a moment. And what it does, like all living things, is it breaks down foods to make ATP, including sugars. Now, the principal sugar in all milk, cow's milk, goat's milk, human mother's milk, is lactose. And what the bacteria first does is it splits this disaccharide lactose into a couple of monosaccharides, including glucose. And then it carries on glycolysis. What's glycolysis? The splitting of glucose in half into 2 pyruvate sugars. But when there's no oxygen, it converts that pyruvate sugar into lactic acid, exactly as we've been describing. Now, in fact, we use this bacteria. You'd say, why would we use this? And people taking lab have learned about this. Because we add this bacteria to milk in order to make yogurt, cottage cheese, sour cream, buttermilk, and kefir. That slightly sour flavor that you taste in plain unsweetened yogurt and in sour cream and in cottage cheese and in an even stronger sour flavor in buttermilk is lactic acid. And a lot of people like the taste of the sour cream and cottage cheese and yogurt, right? So whether you do or don't, many people do. So they like that somewhat sour flavor. But why this is actually even considered beneficial is because some people have a condition which we've spoken of previously called lactose intolerance. They don't have the digestive enzyme lactase to break apart this sugar in milk and other dairy products. So when if somebody has lactose intolerance and they drink milk, it causes them to get cramps and diarrhea. Most of the people with lactose intolerance, however, don't have that problem when they eat yogurt, cottage cheese, and sour cream. Why don't they have that problem? Because there is no lactose in it. Because this bacteria has broken apart the lactose for them into lactic acid. So therefore, they can enjoy yogurt even though they can't have a glass of milk, because there's no lactose in it, because they're just not able to digest lactose. So that's why it's considered a beneficial bacteria, especially if you are lactose intolerant. So that's an example of a bacteria that when it's in an environment without oxygen, as it's breaking apart sugars to make ATP, in an environment where there's no oxygen, it conforms lactic acid. That's called lactic acid fermentation. But this also occurs in muscle cells of us and all other animals. Whenever our muscles, whenever we're exercising, and when we exercise, I'll kind of act it out. Now, what we're doing when we're exercising is we are breaking down sugars with oxygen at a faster rate to make ATP that provides the gasoline, as it were, to power our muscle contractions, to make our muscle cells contract. So the faster you run, the more sugar you burn, and the more oxygen you need to burn that sugar completely into carbon dioxide and water. But sometimes and really commonly, when we're strenuously exercising, very strenuously, we are not able to deliver sufficient amounts of oxygen to our muscle cells. Now, the main limiting factor of what's limiting the ability of our body to deliver oxygen to our muscle cells is not a breathing problem. Assuming you don't have asthma or any other breathing problems, usually most people have tremendous lung capacity and can inhale massive amounts of air, including oxygen. The limiting factor is the heart. The job of the heart is to pump that oxygen in the blood, to pump that oxygenated blood through the vessels of our body to our exercising muscles. And so while all of us, unless we've got a breathing disorder, have plenty of lung capacity, we vary tremendously in the condition of our heart and the ability of our heart to pump very rapidly enough oxygenated blood to the exercising muscles. So as we're exercising, even though we're breathing plenty of air, our heart isn't able to deliver enough oxygen to our leg muscles or the muscles we're using. And when our muscles aren't getting enough oxygen, they cannot break sugars completely into CO2 and water. They can only form lactic acid. It is this buildup of lactic acid in our muscles, when we're not getting enough oxygen to them, that contributes to what's called muscle fatigue and muscle cramping. This is because this lactic acid is just that, an acid and it's starting to affect the ability of the muscles to work. Now, the next page, let's take this one step further. So I have just told you that it's the condition of one's heart that affects his endurance, his ability to exercise for an extended period of time. So we wrote, if you have increased the condition of your heart, if your heart is in fantastic shape, you get the idea? That means that you've got a very powerful and strong heart. It can deliver lots of oxygenated blood to your muscles, and if your muscles are getting plenty of oxygen, they break sugars down completely into carbon dioxide and water and do not form lactic acid. If they don't produce lactic acid, that means you don't get cramps and fatigue. On the other hand, let's assume the condition of your heart's lousy. If your heart is weak, then it delivers less oxygenated blood to your muscles, and when your muscles aren't getting enough oxygen, they form lactic acid. And that buildup of lactic acid creates fatigue and cramping, which means your endurance, your ability to run any long distance is limited. Now, it's really easy to know. It's really easy to know whether your heart is in good shape or not in good shape. If we were to all just start running right now, we're not, but if I said, everybody stand up, let's start running. Some of us, after running 2 blocks, would already be exhausted. That means you're already forming lactic acid. Why? Because your heart is weak and it can't keep delivering oxygenated blood to your muscles. So they're already forming lactic acid. You're developing cramps and fatigue. Some of us could run for 10 blocks. Some of us could run for 1.5 miles. Some of us could run for 3 miles. Maybe a couple of us could run for 10 miles before we get tired. So how long it takes before you get tired and fatigued is a direct indication of the condition of your heart. The way that doctors test this is on a treadmill, and that's called a stress test. Has anybody ever heard of this? They put somebody on a treadmill, they speed it up faster and faster and increase that incline, and they want to see what condition your heart is. The faster they can make that treadmill go, the more they can increase that incline, the harder you can perform without saying, turn it off, I can't take it anymore. That's an indication that your heart's in good shape. If after they speed it up slightly and raise the incline slightly, you're already saying, I can't take it anymore, that's an indication your heart's in lousy shape. All right, now how do you improve the condition of your heart? Your heart is a muscle. If you wanted to make your arm muscles stronger, that means you have to use your arm muscles and make them work. That'll make your arm muscles become bigger and stronger. How do you make your heart muscle bigger and stronger? You have to make it beat faster. Any exercise that speeds up your heart rate is called a cardio workout or aerobic exercise. You probably have all heard that term, doing aerobics, aerobic exercise. Aerobic has nothing to do with lifting weights. What are some examples of aerobic exercises? Running, bicycling, swimming, aerobic dancing, Tai Bo, Zumba, all right? But basically, you've got to do something that speeds up your heart rate. It has nothing to do with lifting weights. Lifting weights with your arms makes your arm muscles bigger, but it doesn't really speed up your heart rate. You doing some press with your leg muscles makes your leg muscles stronger, but it doesn't speed up your heart rate. Only an exercise that makes your heart work harder will make your heart grow bigger and stronger. These are called aerobic exercises or cardio workouts, cardio exercises. They all involve speeding up your heart rate to improve the condition of your heart. So however bad condition your heart is in, and you know it's bad if you can only run 3 blocks and then you're wiped out, that means that you just got to keep pushing yourself further each time. Try to run longer distances. If right now you run 2 blocks, run 3 blocks. And next time after that, run 4 blocks. Keep pushing yourself to run further distances. Keep trying to run at a faster speed. For most people in their 20s, which is most of you, you've got, in order to really make your heart get bigger and stronger, you've got to drive your heart rate up to 160 beats a minute. That's a good cardio workout. So an example of that, whether you're on a treadmill or whether you go to an aerobic dance class. Have you ever been to an aerobic dance class or something like that? A Zumba class, which is very popular right now? That's where they turn on the music and you start doing whatever the hell you're doing. And after 10 minutes, you're dripping wet and you're thinking, surely they're going to turn off the music. Do they turn it off after 10 minutes? Do they turn it off after 20 minutes? At 45 minutes, they're still going. They want to push your heart to its limit. And that's what makes your heart bigger and stronger and improve your endurance. So, one more thing. We have said that whenever your muscles aren't getting enough oxygen, they form lactic acid. Now, that causes cramping and fatigue, as we've been describing. Let me complete this thought. Now, how do we get rid of the lactic acid? We had said this is a reversible reaction. On page H9, we had said on H9, well, we can even see at H10. Let's look at an H10. On H10, so whenever lactic acid is formed when you're not getting enough oxygen, when oxygen becomes available, that lactic acid can be turned back to pyruvate sugar and broken apart when oxygen becomes available. So, how we get rid of lactic acid when we built it up while we were exercising is we have to inhale additional oxygen, additional air when we're done exercising. So how many times have you noticed that when you've exercised very strenuously and you stop, are you like this or are you more like this? If you've stopped running, why are you still breathing at this increased level? Because you have an oxygen debt to pay. That's this term right here on H4. The greater the accumulation of lactic acid, the more additional oxygen that will be required to get rid of that lactic acid when you're done exercising. Because you've got to take an additional oxygen to convert that lactic acid back into pyruvate sugar and break it up completely apart into CO2 and water. That's why you continue to breathe at an increased level when you're done. The longer you're breathing hard is an actual indication of how much lactic acid you accumulated when you were exercising. Has everybody got it? So this is called the oxygen debt. You had a question in the back?
Oh, no.
Okay, no, okay. Now, we had said that there were two types of fermentation. The first type, lactic acid fermentation, is what